very much. Okay, so I'm going to present some updates on CFA molecular breeding. How to do that? Okay, I'm a Mac guy, so sometimes I'm having the difficulty to figure it out. So what I'm going to do that in this presentation, basically on behalf of colleagues from Genomics, Bioinformatics, Biometrics, and our colleagues in the breeding, I'm going to present that what we are doing. So sometimes the first question is that why we need this critical focus area molecular breeding. You know that traditionally our breeders, they are doing great job. However, in development of varieties and the parental lines of hybrid, this takes time. But what genomics can provide that they can help in prediction of the phenotype based on the genotype. What it means that once you have the seeds of the progeny lines, you can just isolate the DNA, you can do the analysis in the lab, and you can predict that this particular line is going to be drought tolerant, for instance, or fusionum wilt resistant, and like that. And if you can improve and if you can enhance the precision and efficiency of prediction of phenotype from genotype, we can transform genetic resources and breeding material into improved germ plants. So that's the reason that we need this CFA molecular breeding, and this cut across the different programs, and in this particular case, grain legumes and dryland cereals. What we need to do, we are having a range of the approaches. I will not go more in the technical aspects in this presentation, but traditionally there are several approaches: marker assisted selection, marker assisted backcrossing. Marker assisted backcrossing is a good approach when you are having the traits controlled by some major QTL or 1 or 2 gene. But if your trait is complex like drought, etc., then we need to use some new approaches called marker assisted recurrent selection, genomic selection, etc. So we need to have the range of the approaches. Sometimes when I discuss with the different colleagues, then they ask, hey, we don't understand what this molecular breeding is going to help or how this is going to help. And I have given an analogy of the molecular breeding with this cartoon. For instance, if you have to reach from Hyderabad to Delhi, this is about 2000 kilometers. You can walk down, but then it will take months. But if you can go by the car also, this needs to take 3-4 days. If you would like to go by train, 24 hours. But if you have the plane, you can go into 2 hours. So these are the different approaches that they can make your life easier. You do not need to travel too long. You can save your time, etc. So what breeders are generally doing in traditionally that you have the germ plant, this can come from the gene bank, this can come from their partners, this is from their developed germ plant, and in the end, they need to develop the superior varieties or the parental lines of the hybrid. Because they don't have the information at the genome level, at the gene level, so we should not blame, but then what happened that without the knowledge, without the genome information, they make a road, but this road is very lengthy, very wavy. Otherwise, ideally, if you have the information, you can reach directly from here to here. But no, you need to reach all these things. Nevertheless, as a molecular breeding, as genomics, what you can do, we can provide the cars to the breeders. So even if you have this long, then this car will run so fast that you will be reaching your destination very fast. So again, this is the next question is that which car you need. Even you are having the variety of the cars. In India, the colleagues from India, they know Maruti 800. Very simple, very robust car. You can use this marker assisted selection kind of approaches by Maruti 800. But if you are having some complex approaches, you need high speed cars. For instance, Honda City or BMW sports car, or sometimes when you are using the approach, the genome wide selection, where you don't need to be worried about one marker, two marker, etc. Here you do the analysis based on the genome wide marker profiling data. You need those Ferrari kind of cars, Formula Race 1. So you need to have all these cars. Next question is, car alone will not be going to help you. Because if we give you the Ferrari cannot be driven from the road from Hyderabad to Delhi. You need to have the highways because this car alone would not work. And sometimes if you are coming from Delhi to Hyderabad, and especially in the month of January or December, you have the weather very foggy, and then roads are also very long, very foggy. So how to reach from this to this? So you need to make the roads clean, you need to remove these fog, etc. And what we have been doing together with the partners from Advanced Research Institute, national partners, during last 4 or 5 years, here we have been investing our time in developing those infrastructure. Now, in several of the cases, we already got the highways. Again, highways will not help you need to have the signpost. Signpost means molecular markers associated with the trait. Otherwise, just genome information, just marker repository are not going to help. You have been given car, you have been given the road, but if you don't have the direction, this is also not going to help. So what I'm trying to say is that through this molecular breeding and CFA molecular breeding, we have to establish all these things. And sometimes we also need to caution that, guys, slow down. You don't need to run very fast, otherwise you may meet an accident. So we need to do all these things. And fortunately, what we have been doing that during last few years, not only the work from ICRISAT, together with several partners, we work on 5 different crops. Sorghum was one of the first crop where we had the genome sequence available. We had large number of molecular markers available, etc. And our colleagues in ICRISAT, led by Tom Hash and other colleagues, they have been making wonderful progress in sorghum molecular breeding. Then after that, we had the pigeon pea genome. Now we had the chickpea genome. And other genomes, they are in the pipeline, pearl millet and groundnut, etc. So in terms of the genome infrastructure, we are not doing bad. Next thing is that those infrastructure, and we are very much grateful to DG and management group that they have been always very helping, and recently we have renovated those facilities at Center of Excellence in Genomics. And in those facilities, we got the genotyping facilities for SSR, simple sequence repeats. So you can consider them like Maruti 800. But nevertheless, you have the different genotyping platform, SNP genotyping platform, DART genotyping platform. Now we got the high throughput sequencing. So you don't need to do the marker genotyping based on the SSR or SNP. Here, at least in those species where you got the genome sequence, you can do the resequencing of the lines. You can have the genome-wide marker profiling data. And some of our colleagues like Punna Ramu has been working very nicely with Ed Buckler in Cornell University, and they are developing the pipeline. So with them, we are also integrating those pipeline in our CFA molecular breeding. And in this laboratory, we don't need to generate just data for ICRISAT colleagues. We are generating the data for outside partners. So for instance, ICRISAT is the blue color, and then national partners in the red color. And you can see that nowadays when we are going, or for instance in 2012, we had a lot of SNP genotyping. At that time, we did not have the facilities. We were outsourcing SNP genotyping outside. But on the other hand, national partner, they were sending their data to their work to us for SSR genotyping. So this shows again another thing that we are generating the interest in the national partners, and we are generating more data for national partners. But now when we got the different machines, we hope that these wars will keep on increasing. So we got those genotyping facilities. Next thing is that tools, databases, decision support tools. And when I'm presenting this, does not mean that this is just Rajeev Varshney's thing, but we are working very closely with Tushar Shah, Abhishek Rathore, and several other colleagues, Rajam, etc., in our computational genomics group. So what we have been doing, that we are having the laboratory information management system. This is being used to generate the data, to keep a track of the data, because we generate not thousands, hundreds of the thousands of data points. So it's very important that we don't create any problems. So all these things, they are very much integrated with the laboratory information management system, which was developed long back, even from the time of Jayashree. Another thing is that earlier we were working in chickpea, pigeon pea, or groundnut kind of crops, and when people would like to move towards the SNP genotyping, we needed to have the SNP markers. In those species, we did not have the reference genome, but again, together with our colleagues here at ICRISAT, with other partners, we developed a pipeline called ISMO, Integrated SNP Mining and Utilization Pipeline. And Abhishek and Sarwar and Tushar did a great work in this pipeline. So what we did, that you can generate, or you can use the next generation sequencing data. You are having the raw reads. If you have reference, this is great. If not, no problem. But by combining these data, and you just put this in the pipeline, because sometimes some of our colleagues, they say, hey Rajeev, now you are talking about millions of the data points. We cannot use that. We say, this is fine. We are developing the pipeline, which will be like a black box to you, but in the end, you will get the lines, that which line you need to use for the crossing. This is our job, that how to make your life easier. So, but anyways, as I said, that you put this data in the pipeline, you will get all kind of information, which kind of, how many SNPs you got, how to visualize them in the different programs, how to use them for the genotyping, etc. So this is one of those pipelines which we recently developed, and we shared it with the International Plant and Animal Genome Conference in this year. Tushar is developing this genotyping data management system. So once you have the genotyping data, you need to put these database somewhere. And we are developing this GDMS. In GDMS, this is good enough for a small to medium scale data set. But if you have to develop, or if you are having larger scale data, we are working with different colleagues in in United States, that how to manage those large scale data, kind of genotyping by sequencing data, etc. But anyway, for our work, for the molecular breeding, marker assisted backcrossing, marker assisted recurrent selection, we are good enough right now and we can use these data. Another tool is that this is molecular breeding design tool, and here this tool can also be used, and this is having the integration with IMAS, which was developed earlier. And in what we can do here, that by using this tool, you can identify which marker I need to use, and when you use the marker, generate the data, put in this pipeline, then this can also select now which line I need to use for making the cross. So you can do this thing, and this is very important. And this is also integrated nowadays with integrated breeding platform, and we call it information system for marker assisted backcrossing. So basically what we are doing, we are developing those tools and we are not reinventing the wheel. If those tools are available, we are just bringing them together. Another thing is, and Dave took very interesting, the very lead role in this system, and basically when we are having the breeding program, you need to have the databases. Some people were using ICIS earlier, but sometimes you don't have those functionality in ICIS, which you need to have the modern breeding. And through initiative of Dave, they have now at ICRISAT we got this AgroBase. And what in AgroBase you can do, that this is a one-stop shop, where you can manage the breeding data, including the management of segregating populations, planting plants, etc. And you can do all these design trials, and Tushar and Abhishek, they are helping the breeders. But one thing what we need to do, that this is for the breeding management, and we are developing the molecular breeding pipelines and tools. They need to have the interfaces. So we are working in those directions nowadays. And we, as I told, that we are part of the integrated breeding platform. In sorghum and millets, we already had several cases where marker have been used in the breeding program, but this is very happy news that even the species like chickpea and groundnut, where we did not think earlier that we can have the molecular breeding product, but now they are also in that field. And in case of pigeon pea, we are using molecular markers or molecular breeding technology for purity assessment of hybrids. So almost in all the 5 crops now, we have started to use the molecular markers in their breeding program. So this is good news. What we would like to do now, as I told, that you need to have the different kind of cars, etc. You can use the marker assisted backcrossing, you can use the marker assisted recurrent selection, we can use SSR marker data, SNP data. But now, as I told earlier, that this is the new approach called genotyping by sequencing, and we are planning to use those approaches. And then, so we want to start the GBS. Second is that we would like to move toward the genome wide selection kind of tool. So we are in process of developing SMO version 2. And here what you can do, that based on the NGS data, based on the GBS data, this will go in the pipeline, and then after the GDMS, this will come here, something will be happening here, and then we will need to provide the information to the breeders. Guys, select these lines for further crosses, so like those kind of things. So basically, don't think that we are putting or generating huge amount of data. Yes, information is huge, but then this will be converted in precise knowledge, and then breeding programs will be basically knowledge-based breeding program. So in summary, I would like to say that, well, so in CFA, unfortunately, we did not have many meetings, etc. But through guidance and the support of DG, we started to have the regular monthly meeting of CFA. We need to have some resources in terms of that management of those kind of information, maybe some human resources, etc. We will discuss later. But so far, what we have been doing, that we are having a state-of-art genotyping facilities. Bioinformatics and molecular breeding pipelines are available or becoming available, and we started to have those regular monthly meeting, not only for the crop people. And this was very important initiative from DG, that now when we have those genomics facilities, they need to help the research program of other disciplines as well, like pathology. We are working with Rajan Sharma, Hari, Gopala, Mamta, so many other colleagues. So like even in case genomics can help in their program, why shouldn't we use them, especially when we have those facilities? So in case some of you are not receiving those mails of CFA, please contact me or my colleague Anu Gorantala, and we will be very happy to have you on the board. And next thing is that now this is the new approach, and we are moving towards using this genomic selection in the breeding program. And we are not working in silo, we are working with the international partners. So for instance, we are very much an integral part of integrated breeding platform GCP. When GCP has sunset, for instance, next year, we need to define the strategy, how to bring this IVP here at ICRISAT, or how to combine these things. So this is a very important thing. Another thing is that we are working with the Gates Foundation, and in January, we had the meeting of Enhancing Genetic Gain, where Dave Washington, Stefania Grando, and myself attended this meeting. And they are also having this big initiative that through in public sector, in CG program, what kind of approaches we should use. that we can have this enhanced genetic gain. And there is another initiative of Gates Foundation, Digital Design for Agriculture. So we are also linking ourselves with them so that again, in the end, we would like to extend in this molecular breeding. One thing is there are several challenging things and we need to discuss. For instance, so far we have been doing this thing at Patancharu level. We need to see, we need to explore the possibilities, how to bring our colleagues from Africa on board, how to reach to those colleagues. So we need to see or we need to discuss those ideas and they are very much in our mind. Okay, so we think that now based on these cars, etc., we can have those roads that you don't need to have the India kind of situation that you are having cars from the both side, but you need to follow those rules, regulations, and you can have the safe driving when you are driving. Don't have the makeup or don't talk on the phone. So we also need to tell these things. So you need to be really careful. And so keep your eye on the road that where you want to reach. So this is very important. So this is not something that you can drive in any direction and you can do whatever you would like to do. In context of the strategic plan of ICRISAT 22 and CGR research program, what we would like to do. So we would not like to continue like this thing with just car. Our plan is to take